Hi, this is Ramya from Bharathasan University. Today I am going to discuss about optical tweezers. Before we are going to discuss about our topic, we should know about what is the history behind the optical tweezers and some basics idea about the optical tweezers. What is optical? We are well known about what is optical. Optical is nothing but relation to the action of light. Here we have to use the high intense light, which means laser light. Tweezers, it is picking up small objects. So optical tweezers is nothing but the arrangements for capturing and moving small particles with laser beams. So first, history of optical tweezers. In 1970, Arthur Askin demonstrated the effect of radiation pressure on lattice spheres in water. In 1986, development of a single beam gradient force optical trap, which means optical tweezers. In 1987, the first application to biological samples, example virtualization. So he was the man who invented the optical tweezers. Next, basics of optical tweezers. Optical tweezers are example of optical trapping, which is a subfield of laser physics. They are used to trap and manipulate particles in the range some micrometer to nanometer and also the measure of the forces. A strongly focused laser beam is used for trapping the particles. The particles that are show dielectric properties. So next, what is optical tweezers? It uses a highly focused laser beam to hold and move microscopic and submicroscopic objects like atoms, nanoparticles and droplets in a manner similar to tweezers. If the object is held in air or vacuum without additional support, it can be called optical levitation. Optical tweezers use light to trap, manipulate and position micron-sized objects. So, optical tweezers is used finding misalignment of atoms or molecules. So, what are the types are available optical tweezers? Single beam optical tweezers, dual beam optical tweezers and holographic optical tweezers. Next, what is the principle and working of the optical tweezers? It is based on the discovery that light has momentum. We are well known about the light has a momentum. Once a laser is encountering an object such as glass sphere or a cell, the light will be refracted. In the center of the beam, the light will be brighter and more light is refracted from here than from the outside of the beam. As a momentum needs to be constant, the object will be trapped in the center of the beam where the forces generated by refracted light is cancelling out. For optical trapping, laser-based optical tweezers are applied which have the optimal properties to generate the optical trap. So this is the light trapping. The first The first image it shows the small transparent spheres are set in a motion when they are illuminated with laser light. Their speed corresponds to Askin's theoretical estimation, demonstrating that it really is radiation pressure pushing them. So the next image it shows one unexpected effect was the gradient force that pushes the spheres towards the center of the beam, where the light is most intense. This is because the intensity of the beam decreases outwards the sum of the forces pushing the spheres sends them towards its center. So next, Askin makes the spheres levitate by pointing the laser beam upwards. The radiation pressure counteracts gravity. So finally, the laser beam is focused with a lens. The light captures particles and even live bacteria and cells in these optical tweezers. So next, Nobel Prize in 2018. So the Nobel Prize in Physics 2018 was awarded with the Arthur Askin. Askin focused laser beams with lenses to create optical tweezers. The radiation pressure produced by the light holds the particles in place. Combining optical tweezers with other methods that stop and trap atoms allows visualizing of molecules that perform important roles in cells. Some examples of early single molecule applications. So, in his initial experiment, an optical forces as can observe something unexpected. Latex microspheres near a weakly focused laser beam was drawn in the form of periphery of the beam toward the beam's central axis. A ray of light is bent by refraction when it passes through a transparent sphere, and because light carries a momentum. This imparts a force onto sphere in an asymmetric optical field. The sum of all such rays results in a net gradient force in the direction of higher light intensity. Askin would go on to create a single beam gradient optical trap or optical tweezers. So, next, it was tracked the motion of an optical trapped sphere and it was pulled by a single kinesin motor walking along a microtube. This revealed that kinesin walks along microtubes in 8 nanometer steps and can generate 5 micrometer of the force. So the next image it shows is use a dual optical trap to determine the mechanical results of ATP hydrolysis by a single myosin molecule. They found that each hydrolysis even corresponds to an 11 nanometer power stroke accompanied by the generation of the forces. So next, it is used optical tweezers to study the elastic properties of DNA and the behavior of DNA under high forces. So the final image it shows is measured the force generated by RNA polymerase during transcription and studied the motor's behavior under varying mechanical loads. So what are the applications of optical tweezers? Cell-based studies, cell interactions and intracellular manipulations, measurement of binding forces, bacterial and viral adhesion studies, molecular motor studies, actin and myosin, kinesin and dynein motors, DNA or RNA binding, interaction forces, helicase and translocase activities, lab on a chip device, which means used to biosensor assays. So thank you so much.